Hi guys, and welcome to this lecture video on reproduction. We're going to talk about sexual versus asexual reproduction and the differences between mitosis and meiosis. Okay, so in sexual reproduction, some advantages of sexual reproduction are enhanced evolutionary fitness. You're going to be more fit, and there's a few reasons why. The first reason is because you have a varying genetic makeup. So you are not going to be identical to your parent. You're going to be different, and that means that you're going to be more fit in the population because of this varying genetic makeup. Also, it rids population of harmful genes fast. Because you're not identical to your parent, then we can actually get rid of harmful genes much quicker. And if you look over here, you can see you have the egg and sperm, which would be fertilized. And then in sexual reproduction, you're going to go through the process of meiosis, which we'll talk about a little bit more. The last thing I want to mention on this slide is that notice that the zygote is what we call a diploid. And so it has 2N or 46 chromosomes here, and the sperm is actually haploid, and so is the egg. So we have a sperm that has 23 chromosomes and an egg that has 23 chromosomes. And when that sperm fertilizes the egg, that creates our zygote, which has 46. So when you think haploid, just think half the number of chromosomes. So a sperm cell would have half the number of chromosomes that the individual that created that sperm has. Okay, so advantages of asexual. A in front of sexual here means without. And I always say for asexual reproduction, no egg, no sperm, no fun. So asexual reproduction does not require egg or sperm. This is really good and useful for an individual when finding a mate is hard. So when it might be hard for you to find a mate, being able to reproduce asexually is advantageous. Also, it eliminates energy used to form gametes and copulation. So we're going to take up energy. It's going to require energy for us to create those sex cells, the egg and sperm, or those gametes. Gametes is just a fancy term for egg and sperm. And so it's going to take energy to create those. It's also going to take energy for copulation or intercourse. So in asexual reproduction, we don't actually have to require that energy for intercourse. So, all right. Cool thing about asexual reproduction is the daughter cells or the resulting cells are identical to the parent cell. I want to show you an example. Down here, you can actually see the C star, and it's doing what's called fragmentation. There's many different types of asexual reproduction. You have fragmentation, budding, spores, lots of different types of asexual reproduction or methods or mechanisms of asexual reproduction. But this is fragmentation, and what that means is this C star actually can release one arm. So you can actually see it's released this arm here, and from that arm we're growing a completely new C star over here. And then the arm here can actually regrow on the C star, which is pretty cool. And this C star will be genetically identical to the parent over here because it's doing asexual reproduction. All right, so let's talk about the difference in mitosis versus meiosis. So mitosis is what we're going to use in asexual reproduction, and meiosis is in sexual reproduction. Mitosis is also the process when you're like 7 and you fall and scrape your arm because you were riding your bike, and new cells grow back. That's also the process of mitosis. But here I want to talk about reproduction. And so mitosis is going to be used by individuals that reproduce asexually, and meiosis is going to be used by organisms that reproduce sexually. And so there's a few key differences between the two. In meiosis, we have, this is what we do, so let's look over here first. We have daughter cells that are going to be haploid. Basically, that means our sex cells, or egg and sperm. They're going to be haploid. So they're going to have half the number of cells than our normal cells, like your cheek cell. Sorry, half the number of chromosomes, excuse me, that your cheek cell would have. Because we need 23 and 23, and when they fertilize, we end up with 46. So I'll kind of show that over here. So here's your egg with 23 chromosomes. Here's your sperm. It's a terrible drawing. I'm not an artist. And then it'll unite and we'll end up with a fertilized egg with 46 chromosomes. So that's why these daughter cells have to be haploid or have half the number of chromosomes. So this fertilized egg here is going to be genetically different than the parent, because we're taking part of dad, right, and part of mom and putting them together to fertilize the egg. So we're going to end up with a genetically different individual. You're going to have some characteristics that you get from your mom, some characteristics that you get from your dad, and you're going to end up being genetically different. That's because of a process called crossing over. So crossing over is going to occur within the chromosomes in this process of meiosis. It's a little bit more complicated, and I'm not going to talk about exactly where crossing over occurs, but you can look in your textbook if you want to reference more about crossing over. But basically, the chromosomes cross over, and you end up with a chromosome that has part of your mom's information and part of dad's, and that leads to being genetically different from them. And then it's also going to produce 4 daughter cells. So we're going to end up producing 4 sperm and 4 eggs. However, only 1 egg is going to be viable, and the others just disintegrate. So that's meiosis. Let's go back over here to mitosis. In mitosis, we're going to have daughter cells. We're going to end up with 2 daughter cells that are genetically identical to the parent. That's because no crossing over occurs. And because they're genetically identical to the parent, we're going to have the same number of chromosomes as the parent cell. So let's say we have an organism that has 40 chromosomes. In mitosis, you're going to end up with 2 daughter cells, 2 new cells that are both going to have 40 chromosomes in them, because that's the number that the parent started with. So hopefully you're starting to see some differences here between mitosis and meiosis. All right, so why sexual reproduction? Well, we need offspring, right? To pass on our genes. We need offspring to carry those genes from generation to generation. And offspring are going to get genes from 2 different parents, so they're not genetically identical. This is actually a really good thing, because sometimes there are slight mutations in genes that can cause you to be less susceptible to a certain disease. If you reproduce asexually, all of your offspring are going to be identical, and a disease might wipe out. You might be susceptible to a certain disease, and an entire population can be wiped out quite quickly. So being genetically different is advantageous. Research has shown that most organisms reproduce sexually just because it's their instinct. They don't get any pleasure out of sexual reproduction, except for the dolphin. So the dolphin is the exception. Dolphins and humans will perform copulation, if you will, or intercourse, even without the purpose of reproducing. That's kind of the definition of for pleasure, if you will. They'll reproduce without or copulate without the intention of reproducing. All right, so let's look at some mating and courtship behaviors. Some of these are really, really cool. So here, this is the giraffe. You can see that this guy here, this is the male, and this is the female. And I know that because the male is back here, and he is actually tasting the female's urine to see if she is fertile. And so if it has a certain taste, the male will know that she's fertile and that it is time for him to copulate with her, which is kind of cool. You might actually see this happening. I took a group of students to the zoo once, and it was actually happening right in front of us, which was awesome. So they knew exactly what was going on after we went over it in class. But you might actually see this if you take your kids or, your significant other, or you go to the zoo or whatnot. You might actually see that, which is kind of cool. I like this guy down here. So this is a bowerbird, and unfortunately he is using straws and plastic bottle tops to create his nest here. But with bowerbirds, the male that builds the largest, best nest is the males that the female would like to mate with. So she's looking for a guy that can really support her, a guy that can build a nice big nest for her, and that's the one who ends up winning the female. So here you can see he's building a nice nest. Unfortunately, though, it's got lots of plastic bottle caps and whatnot, which we as humans are terrible for, not recycling and things like that. Anyways, we'll move on. This one's pretty cool. Flatworms. So I'm probably gonna guess that you've never heard of penile fencing. But flatworms, this is the bottom of the ocean actually. So this is the bottom of the ocean. And these flatworms are doing what's called penile fencing. And they are hermaphrodites, which means they can be either males or females. And to kind of determine this, they are fencing here with their penises, and whichever one jabs the other one first and injects its sperm into the other one will end up being the male, and the one that gets the sperm injected into it ends up taking on the role as the female and having to take care of the offspring. So anyways, you want to win this fencing competition basically, so that you are not stuck with raising the offspring, if you will. Down here we have a peacock. You're probably familiar with these. Basically, the females or peahens are looking for the peacock that has the brightest, most colorful feathers. Unfortunately, if you are an albino peacock, then the females are less likely to mate with you. Also, if your feathers aren't bright and colorful, maybe you're not getting certain nutrients in your diet to make your colors, your feathers bright and colorful, then you're also less likely to get a mate. And the last one I have saved here for you is Anachinus. Anachinus is a rodent. I actually think it happens to be a really cute rodent. But what's interesting about the mating or courtship behaviors of Anachinus is the males are pretty much players. So the males kind of like run around, scurry around, and try to mate with as many females as possible. They're pretty aggressive, actually, towards the females, and they just run around for 2 weeks solid. Okay, so it's like a party. For 2 weeks, they run around and try to mate with as many females as possible. Sounds like spring break, right? Anyways, just joking. Sorry. Okay. Moving on. Basically, what happens after those 2 weeks, though? Remember how we talked about how sexual reproduction requires a lot of energy for copulation? Well, this guy has passed on his offspring, provided as many chances or opportunities to pass on his offspring as possible. But at the end of this 2 weeks of like intense copulation, he actually dies because he uses so much energy in copulation. He won't remain alive, which is kind of neat, I think. Anyways, so these are just a few mating courtship behaviors of organisms. Okay, so here we're going to talk about male reproductive anatomy. So the primary sex organ here is the testis, which you can see here. And then there are secondary sex organs, penis, ejaculatory duct, seminal vesicle, etc. A few things to point out. The seminal vesicle, which is located here, is going to produce the semen, and the sperm is actually made down in the testis. So the testis is where the sperm is going to be made. It goes into this structure called the epididymis, which is the mature sperm warehouse. And then it leaves the warehouse here in the epididymis via the vas deferens. And so I think it's a big misconception that the sperm just travel up and out. But actually the vas deferens go all the way around the bladder and meet the seminal vesicle here at what's called the ejaculatory duct. And notice that there's this purplish structure here, which is the prostate gland, and there's going to be some prostatic fluid as well that is going to mix with the semen coming from the seminal vesicle and the sperm that's coming from the vas deferens at the ejaculatory duct, and is going to be ejaculated, and that's what we call ejaculatory fluid. Now, there's this other structure here called the bulbourethral gland, which you can see is right underneath the prostate, and that's going to secrete pre-ejaculatory fluid. And the pre-ejaculatory fluid does have a purpose. So one has two functions. The first function is lubrication, and the second function is actually to buffer the pH of the male urethra. So if you look at the male urethra, you can see it's actually quite long, and you can see this is the bladder here, and so the ejaculatory fluid is going to move through the urethra, and so is the urine coming from the urinary bladder. And so this bulbourethral gland will secrete pre-ejaculatory fluid, which has a different consistency than ejaculatory fluid. It is a clearer and not as thick substance. And what it's supposed to do is actually buffer the pH of this urethra so that the sperm can make it through the urethra without major changes to the pH. So the pH can affect how the sperm react. And so it's basically a buffering fluid, which is pretty cool. To note another structure, you have your corpus cavernosum, and this is the structure that is going to fill with blood to cause an erection. All right, so now that we feel comfortable with male reproductive anatomy, let's talk a little bit about a female. So the primary sex organs, or where the eggs are going to be made, is in the ovaries. So you have two. There's only one pictured here, but you have two. So here's the ovary. A few other structures. You have the fallopian tube here, which is actually, or uterine tube. You can use either term, but that's actually where the fertilization occurs. So it actually occurs in the uterine tube. So what's going to happen is the sperm are going to have to go through the vaginal canal, up through the cervix here, into the uterus, and then they're going to have two options. They can go into the right fallopian tube or the left fallopian tube. And in most cases, there's only one egg released, and so half the sperm swim into the empty fallopian tube and half the sperm swim into the fallopian tube containing the egg. And it is the first sperm to fuse its membrane with the egg that will actually fertilize the egg. And so what happens is the egg actually makes its way back down into the uterus where it implants itself. So it's fertilized in the fallopian tube or uterine tube, but then moves itself back down into the uterus where it'll implant itself and grow in that rich uterine lining until it becomes into an embryo and then into a fetus. Now, if you notice something about the uterus, so this is where the fetus will be housed in the uterus here. If you look down, you actually see that the uterus sits on top of the urinary bladder. And so that's why a lot of females, when they're pregnant, they have to urinate often, because the uterus is pushing down on that urinary bladder. A few other things to point out. You have the labia minora and majora. These are the they're basically flap-like structures that open into, and you can see into the vaginal canal, and also there's the urethra. And then we have a structure called the clitoris, and it's going to be a stimulating structure. So in the males, I think a lot of times people kind of associate, like, what's the equivalent of a penis in a female? It would actually be the clitoris, because they're both stimulating organs, which is great, I guess. Okay, moving on. All right, so sex determination in humans. All eggs are going to carry an X chromosome. So all the eggs carry an X, and it's the sperm that's going to determine the sex of the individual. So if this sperm is also carrying an X chromosome and they meet, then we're going to end up with a female. if we have an egg, which always has an X, and a sperm that's carrying a Y chromosome meet, we're going to end up with a male. In sea turtles, though, it's very different. So sexual determination in sea turtles is based on the incubation temperature. How cool is that? Wouldn't it be awesome if you could like change the, like determine the sex of your child by like cooling yourself off or heating yourself up? I think that's kind of neat. But basically the incubation temperature here determines the sex in sea turtles. So at higher temperatures, about 85 degrees Fahrenheit or hotter or warmer, then we're going to end up with females. And if it's about 82 degrees, we're going to end up with more males. Now, in a clutch of about 100 eggs, then you're going to have about a 90 to 10% ratio. So if the nest is incubated at about 85 degrees, you're going to end up with about 90% of that clutch being female and 10% male. So it's not an all female clutch or an all male clutch. It's just about a 90 to 10% ratio most of the time. All right, so sometimes in humans, meiosis kind of can go AWOR or a little crazy, and sometimes our chromosomes do not split correctly. And so with Klinefelter syndrome, this is going to be a male, but we're going to have two X's instead of just one X and a Y. We're going to have two X's and a Y. And that's going to be a non-disjunction. That's what the ND stands for here. We'll talk more about that when we get to genetics, but basically the chromosomes didn't separate correctly during egg or sperm formation. The frequency of the population is about 1 in 2,000 here for males. And that male is going to be sterile and also have feminine characteristics like enlarged breast. For example, you can take testosterone as a treatment for Klinefelter syndrome. If you end up with XYY, that's going to be a normal male. Same frequency in the population, it's just involved in sperm formation. XXX, you can have 3 X's, you would not know it, but because you would end up with a normal female, and that's about 1 in 1,000 people that have 3 copies of the X chromosome. And then Turner syndrome is the only case in which you can have 45 chromosomes in a human and survive. So here you have an X, and this O is a placeholder for the Y. This is involved in egg and sperm formation, and it's about 1 in 5,000 people that have Turner syndrome. All right, so the things that you see on this slide can actually cause you to have difficulty reproducing. Now, please do not think that the things on this slide will prevent you from having an offspring, okay? That's not the case, all right? So don't partake in these and think you are good to go. You're not going to have any children because that's not the case. So, but if you're wondering why fertility might be an issue for you, then here are some reasons. And it's not always these, but these can cause you to have difficulty reproducing. Excessive use of alcohol. Excessive, so that doesn't mean just, you know, a beer on the weekend or something like that. I'm talking about like alcoholism, excessive use of alcohol can cause you to have difficulty reproducing. Obesity can also cause issues. Smoking as well. And here, marijuana, actually, it will cause the sperm to speed up their like swimming action, and they'll actually tire out too fast before they reach the egg. That's why you might have issues reproducing if you are partaking in marijuana. And the most common reason actually is for males is actually the use of a sauna or a hot tub. So a friend of mine had issues reproducing, and they went to their doctor, talked to them, and they asked, like, you know, are you getting in hot tubs? Are you going to the sauna? What's going on? And the guy was like, oh, well, we just bought a new hot tub. And they, every afternoon after work, they would like sit in the hot tub to relax. And so the doctor was like, you know, why don't you get out of the hot tub, stay out of the hot tub for 3 months and see if that helps. And so they ended up actually getting pregnant after not using the hot tub for several months. And one of the reasons why is the testis does not like warm temperatures. And remember, the testis is where the sperm is going to be produced. So as we heat up the testis, we're actually going to decrease sperm production. And so if you already have a low sperm count, then these things are definitely something that you're not going to want to do if you are trying to reproduce. All right, methods of contraception. There are many. So we have barrier methods of contraception. We have female, male condoms. We've got the sponge, diaphragm, etc., etc. We also have behavioral, so abstinence, and also you can look at the days, look at the cycle, and actually 20 through 28, you're infertile. And so you can actually look to see when you would be infertile for behavioral methods. There's also the withdrawal method that some people use, or rhythm method. Then we have hormonal. So we have the patch, we have the NuvaRing, we have the pill, and also the implant as well. That is hormonal. And then you can have surgical. So here you can have tubal ligation, or basically the tubes are tied so that the sperm can't get to the egg that's being released. Or you can have a vasectomy, and in a vasectomy, they actually cut or burn apart the vas deferens here. However, it's not wise to get the vasectomy as much, because that tissue, the vas deferens tissue, is actually known to grow back quite a bit. So if you do get a vasectomy, you would need to go back and be tested to make sure that it has worked properly and that the tissue has not grown back, because it is likely to grow back sometimes. Okay, so here are failure rates for contraception, just to get you familiar with some protection or these contraception methods. I would probably ignore the perfect use column here, because I doubt anybody actually uses their method of contraception perfectly. So let's just focus on this typical use column over here. So this is pregnancies per 100 users. So basically the numbers that you see here underneath typical use are the percentage likelihood of getting pregnant. And if you do not use any protection, you have about an 85% chance of becoming pregnant. If you do the rhythm method, that would be 25% withdrawal. So if you do the withdrawal method, that's 27% chance of pregnancy. The male and female condom, that's about 15. Even with the condom, there's about 15 to 21% chance you can still become pregnant. The pill, patch, and NuvaRing are about 8%. This here is the shot, and that's 3%. An intrauterine device is actually less than 1%, and so is tubal ligation and a vasectomy. So let's talk about types of STDs. I'll start off with the one up here at the top. So this is crabs, not this guy here, although he's really cute. This one probably not so cute. If you found this one on you, you're probably not as excited about it. There's two different main categories of STDs. You have bacterial STDs and viral STDs. And if you go to the doctor and your doctor says you have gonorrhea, chlamydia, or syphilis, you should be ecstatic. Really excited about having gonorrhea, chlamydia, or syphilis, because all three of those are bacterial and they can be cured with antibiotics. Yes, you can get rid of syphilis, chlamydia, and gonorrhea. These STDs underneath the viral column, genital herpes, genital warts, hepatitis B and C, HPV, HIV, and AIDS, if your doctor lets you know that you have any of those. Unfortunately, because they are viral, they will stay in the body, and what we can do is actually reduce the symptoms, but we can't completely get rid of any of the viral STDs. So that's why I said you want to be excited about having the bacterial STDs. If you do have, get tested and your doctor's talking with you. All three of those are reasons to get excited. These over here, not so much. And a lot of these, like genital warts, for example, you may actually contract the virus but not show symptoms for 3 years. So you may not actually have the first wart form for 3 years after you have contracted the virus. And what's happening then is any of your partners that you had during that time, you have actually transmitted the virus to them, not knowing that you have it, right? Because you don't have any symptoms. You don't have any warts, so you don't think you have it. But then you can actually you're actually transmitting it to other people. So it's important to get tested. And then lastly, I love this photo. It was taken by Eric Snyder, and this is the male here, dragonfly here, and you can see he's releasing sperm into the female, and the female is actually holding on to the male, basically the back of his head or his neck. But if you look at it sideways, you can actually see a heart form when they copulate, which is kind of cute.